We are starting with a new portion named glycolysis. Now, what is glycolysis? Glycolysis, we can easily break the name or the definition of glycolysis, which means glucose breakdown, or to break down glucose is known as glycolysis. Now, this glycolysis, it takes place in both aerobic and anaerobic respiration process. Now, this glycolysis, it takes place inside cytoplasm. It is also known as EMP pathway. Why it is known as EMP pathway? It is known as EMP pathway because it has been given by three scientists, Emden, Meyerhoff, and Parnas. So, from the name of the three scientists, we have given the name glycolysis, the name EMP pathway. Now, glycolysis, it's a bit complex process in which one glucose molecule gives two molecules of pyruvic acid. One glucose molecule gives two molecules of pyruvic acid. This particular process glycolysis is dealing with is entirely on this one. Now we will be seeing stepwise how this particular process takes place. The names of the enzymes as well as the names of the and the steps where ATP and ADP conversions are taking place, that one is important. So I'm keeping this portion empty so that finally we can keep a note of the entire process, and I'm taking a fresh page for this conversion or this entire process of glycolysis. First, glucose is converted into glucose 6 phosphate. Glucose 6 phosphate. So to convert this 6 phosphate, now when we are adding phosphate, we need to remember that if we break ATP, it will give ADP plus inorganic phosphate. This phosphate will bind over here. So over here, what is happening? ATP is getting converted to ADP so that a phosphate group is released which binds with this glucose to give glucose 6 phosphate, and the enzyme responsible is hexokinase. Next, this glucose 6 phosphate, it converts into fructose 6 phosphate by the presence of the enzyme phosphohexoisomerase. Now this fructose 6 phosphate is converted into fructose 1,6 phosphate, and the enzyme, first we will see that it is again that ATP is converted to ADP because phosphate group is one more phosphate group is added. So the enzyme responsible is phosphofructokinase. Phosphofructokinase. Now, this up to here, we will be calling this process, not only up to here, we will be seeing that this fructose 1,6 phosphate, it is 6 carbon compound. Up to here, all of them are 6 carbon compound. Now, this particular compound will be breaking into two compounds having 3 carbon each. Will be breaking into two carbon compounds having 3 carbon each. One is DHAP, another is PHAL. DHAP is dihydroxy acetone phosphate, and this one is phosphoglyceraldehyde or 3-phosphoglycerate, or we can say 3-phosphoglycerate or 3-phosphoglyceric acid, or we can also say phosphoglyceraldehyde. They can interchange into one another. Now to break this one, the enzyme involved is aldolase. And to convert this to each other, the enzyme involved is phosphokinase isomerase. This enzyme, phosphokinase isomerase, is responsible for reversible conversion between this DHAP and phosphoglycerol aldehyde. Now this phosphoglyceraldehyde, this DHAP, it converts over here. So when it is converting to this, we are having 2 moles of phosphoglyceraldehyde. This one converting to this one, we are having 2 moles of this one. So we will be proceeding further with only phosphoglyceraldehyde. Now the enzyme that is responsible is known as glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase, and and in this process what happens, NAD plus converts into NADH2. It forms 1,3-phosphoglyceric acid. This one is formed in this process. Now we will see there's a decrease in the number of carbon or phosphate. It will form 3-phosphoglyceric acid. Now this time ADP will convert to ATP. Why? ADP plus phosphate gives ATP, so that we are taking one phosphate from here, adding it to ADP to form ATP. Now this one is known as substrate level phosphorylation. What is phosphorylation? When we add phosphate group to ADP, it forms ATP, and as from the substrate level we are collecting the phosphate to form this one, it results in the formation of this particular compound. So 3-phosphoglyceric acid, it takes place in the formation in presence of the enzyme phosphoglycerate kinase. Now this one converts to 2-phosphoglyceric acid. So 2-phosphoglyceric acid, it takes place in the presence of enzyme phosphoglyceromutase. Now in presence of enolase, what will happen? Water will come out, resulting in formation of phosphoenol pyruvate. This phosphoenol pyruvate, again ADP is forming ATP, and it forms pyruvic acid. Now keep in mind that I have already said that it is converting to this one. So 2 mole. 2 mole means everything we will multiply by 2. That means 2 ADP is forming 2 ATP. 2 NAD plus is forming 2 NADH2, and 2 ADP is forming 2 ATP. That means in this particular step, total 2 ATP is formed. Total 2 plus 2 ATP is formed. That means 4 ATP is formed. And in the previous one, what happens? Only 280 ATP is utilized in formation of ADP. That means 2 ATP is utilized. So from this entire glycolysis process, we say that the net result or the net income is 2 ATP. We need to see this entire process, and this is what we call glycolysis. Stepwise, if you remember the enzymes and right, we can complete this glycolysis.